Okay, guys, I'm going to just do one last recording to help you get through chapter 9, muscles and muscular tissue. I'm just going to hit on a few of the topics that I wasn't able to get to in my first two recordings. So the first thing I'm going to call up a whiteboard here. I don't have my eye pencil with me, by the way, so the writing on this is going to look weird. It's going to look really thick until my eye pencil charges. Anyway, I just wanted to show you maybe this won't work. Let's see. It's supposed to be an E. The repeating pattern of myofilaments. This is all in your book. We already know all about myofilaments, actin and myosin, and a little bit about titin. So what I'm going to do is just draw a quick picture for you. I'm going to draw one sarcomere, and you should know what that is now. That's one contractile unit of overlapping myofilaments. That's the M line, that vertical line, and then these transverse or horizontal lines would represent myosin. So I draw 3 of those. I'm not going to draw the myosin heads on there, those little club heads, just to simplify this, because that's not necessary right now, but they do exist. This is a typical schematic drawing that textbooks and scientists / anatomists use to show the overlapping myofilaments, the pattern, the repeating pattern. So there's these horizontals are myosin, the vertical is the M line. Now I'm going to draw the actin molecules, and those typically look something like this. I'm going to label this piece. What I've drawn for you here is just one sarcomere. One contractile unit basically goes from this, it's called the Z disc. You can see why, because it looks like a Z. That's the Z disc. This is actin, the yellow horizontal lines. This is myosin. This is the M line. And one sarcomere goes from one Z disc to another that includes basically 2 sets of actin molecules and technically 2 sets of myosin molecules, one on this side and one on this side. Now, this pattern repeats thousands or hundreds of thousands of times down a myofibril. And so now I'm going to just draw just to sort of accentuate that point. These would be more actin molecules that I've just drawn. Then there would be a new set of myosin molecules here, another M line, another set of myosin. The point that I'm trying to make here is that it repeats over and over and over again, all the way down, hundreds of thousands of times down a myofibril, that specialized organelle that contains these myofilaments. And it just keeps going. Then we would have more of these actin molecules, yada, yada, yada. So you get the idea. Now, there are a few, the overlapping patterns of actin and myosin. Sorry, let me get a different pencil so you don't think that's an actin molecule. Here they overlap, here they overlap, here the actin and myosin overlap. All of these zones of overlap or non-overlap have names, just sort of have names given to them. So I'm gonna clean up my drawing really quickly and do some erasing now that you know what things are. I'll leave Z disc on there. I'm just gonna get rid of this because we know that that's myosin. there. Now, right between here and here, where there's no actin molecules, we call that the H zone. There's just names to these overlapping areas. We know that the M line is in the center. Where there is from the tip of a myosin molecule out here, right there, to the tip of the other myosin molecule, which would be right there, that's called the A band, capital A, and that's just where there is myosin, B-A-N-D. And then there's a Z disc, I already labeled that one, where there is no myosin, so from right here to this area, where there is no myosin, excuse me, we call that the I band, capital I, B-A-N-D, sorry about the writing, I'm trying to write small with a very large pencil tip. Finally, that's all I want you to really know. One sarcomere, the A band, the I band, the M line, the Z disc, the H zone, and I'm going to star the H zone. The H zone specifically is the area within a sarcomere that shortens, narrows, and eventually disappears as the actin molecules get pulled by actin toward the M line. So you know that when a muscle contracts, the myosin head reaches up, grabs actin, and pulls actin in this direction. And it pulls all the actin molecules on this side toward the M line, all the actin molecules on this side toward the M line. So this area, the H zone, during contraction, it shrinks. It gets narrower from actin going this way and this way. And sometimes it even disappears. The actin molecules come all the way to the M line here and all the way to the M line here. All right, enough of that business. Well, before I erase this, that third myofilament, titin or elastin, just to show you how it's attached. It's this, I'm going to draw a slinky coil, like a spring coil. It attaches from the end of myosin actually to that Z disc like this. So think of this as an elastic band that when a muscle gets stretched out, not when it contracts, when it gets stretched, these coiled springs, they get stretched like this. And then when the muscle is no longer stretched, this recoils because it's elastic back to its original shape. All right, now that's enough of that. Let me see if my iPen is charged. We'll get on to a couple last topics, and then we'll call it good for chapter 9. So I believe I went over isotonic and isometric contractions, the difference between the two. I'm just going through my notes to make sure that I went over everything. We did muscle metabolism, a myoblast. I do want to talk about force of muscle contraction. So I'm going to call it my new whiteboard. Force of muscle contraction. It's called contractile force. When a muscle contracts, it creates force, tensile force. Sometimes we call it tension as well. That's just the energy that's created when a muscle contracts. Now, the force of a muscle contraction, how strong it is affected by 4 factors. I'm just going to write affected by, and I'm going to list these as 1, 2, 3, 4. The first is the number of fibers that get stimulated during contraction. We've talked a little bit about this. The more fibers that get stimulated, the stronger the contraction, the more force is produced. That's pretty logical and intuitive. The second factor affecting contraction force is the size of the fibers that are stimulated. Smaller fibers produce less force than larger fibers. Number 3, the frequency of stimulation. That can be additive. Just think of the temporal summation that we talked about last time. The frequency of stimulation, that refers to the neuron or nerve cell. So the faster, the higher rate that the nerve cell stimulates the muscle fiber, the more contractile force is produced. And finally, number 4, this is probably the least intuitive. The degree of muscle stretch. What does that mean? That means that there is an optimal length of each muscle for each muscle prior to contraction that will produce the greatest force. So I'm just going to write in parentheses, there's an optimal length which produces the greatest force. Think of it this way. If a muscle is too stretched out before it starts to contract, it can't produce as much force, nor is if it's already halfway or mostly contracted. Those myofilaments are so overlapped, they have a hard time getting going. So there is a perfect, I don't want to say perfect, there is an ideal length, degree of muscle stretch prior to contraction that will produce the greatest force. Not sure I mentioned these 2 terms. I believe I did, but just in case I didn't, I think I did, but I teach so many A&P courses. Sometimes I forget what I told which sections or which classes. Hypertrophy, I do want you to know hypertrophy just means the increase in muscle size due to activity. Think of weight training or resistance training. Atrophy is actually the decrease in muscle size or loss of muscle tissue due to inactivity. So let's talk about that. And I know I went over the different types, the 3 types of muscle fibers: slow oxidative fibers, fast glycolytic fibers, and fast oxidative fibers. Within those 3 fiber types, a muscle is typically made up of a mixture of all 3 of those fiber types. It's not just one of those 3 that makes up an entire muscle. We all have all 3 types in our bodies. The difference between us as individuals, some individuals have more of one type than others, but we all have the same muscles, and we all have all 3 fiber types within our bodies, but sort of the proportion of those 3 muscle fiber types varies a bit from individual to individual. Last but not least, I want to say a few things about smooth muscle. So we're going to get off skeletal muscle. This is, I believe, at the end of the chapter. Smooth muscle is a whole different animal than skeletal muscle. Up until this very moment, 99% of what we've talked about has been skeletal muscle, the stuff attached to our bones that we have voluntary control over. There are multinucleate cells that have their own nerve innervation, yada, yada, yada. Smooth muscle, I said a few general characteristics about it at the beginning of the very first lecture, but I'm going to give you some more detail about that. These are spindle-shaped cells. I'm just going to write spindle-shaped. Think of them more like this. Skeletal muscle fibers that we've talked mostly about, they're long and cylindrical like this. That's a smooth muscle, spindle-shaped cell. Remember, they are not striated. They don't have stripes under a microscope. They're involuntary. That means we don't have conscious control over them. They have 1 nucleus per cell. All of this was written out in the first lecture. And their location is they line visceral organ walls and blood vessel walls. So here are some similarities and some differences of smooth muscle, some characteristics. The cells, the individual fiber cells, have, actually the entire muscles, the sheets of muscle, have endomysium covering only. So remember, in skeletal muscle, there's endomysium, perimysium, and epimysium that makes up the entire muscle. In smooth muscle, the cells only have endomysium wrapped around each individual cell. There is no perimysium. There are no fascicles or bundles, and there is no epimysium. Of the tissue, the smooth muscle tissue, there's typically 2 layers involved of tissue. So when this muscle tissue is lining visceral organs like our stomachs and our stomach and our intestines and our trachea of the respiratory system, they are in long flat sheets wrapping around those organs, and there's usually 2 layers of tissue. And there is a longitudinal layer and what we call a circular layer. Those names just refer to the direction. That's a U, circular layer. Those names just refer to the direction that the fibers run. So if on the outside, for example, of a blood vessel, not on the outside, in the covering, in the wall of a blood vessel, we have smooth muscle. The longitudinal layers, the cells, the fibers, those spindle-shaped cells, they run this way, long, lengthwise. The circular layer of tissue runs this way, around the diameter of the vessel. So there's usually 2 layers of smooth muscle tissue. The stomach is something unusual. The stomach has 3 layers of smooth muscle tissue. There's an added oblique layer of smooth muscle tissue in the stomach. Reason for that is the stomach has so much mechanical digestion responsibilities to do, so it has to really churn and crank out contractions. Some of the actions that smooth muscle produces when it contracts are peristalsis in the GI tract, the digestive system. That just means the waves of muscular contraction that propel food along the GI tract. And vasoconstriction in blood vessels. When smooth muscle contracts, the diameter of our blood vessels gets smaller. When it relaxes, the muscle relaxes, that increases the diameter of the blood vessels. Smooth muscle is under the autonomic nervous system control. So autonomic NS control. We haven't studied the nervous system yet. This is the part of the nervous system that we don't have conscious control over. The neurons, the nerve cells, are these bulbous, misspelled that. B-U-L-B-O-S are bulbous swellings. And those are called varicosities. So if you see that word in the chapter that you're reading, varicosities just refers to the ends of these neurons that innervate smooth muscles. Varicosities release the neurotransmitter, but they usually aggregate and occur in these bulbous swellings, and they diffuse that neurotransmitter over a very wide area, a sheet-like area. Smooth muscle cells, there are no T-tubules, transverse tubules, inside of the cells like there is in skeletal muscle. The cells have calcium channels that allow calcium to influx from outside of the cell. I'm going to write for influx from extracellular fluid. So let's just think about this for a minute. If there are no T-tubules inside of smooth muscle cells, that's where calcium is stored inside of skeletal muscle, but there aren't any of those. So we have to get calcium from somewhere, and calcium, the bulk of the calcium, not all of it, but the bulk of the calcium in smooth muscle contraction comes from outside of the cell. So there are openings, channels, calcium channels. I'm just going to draw the nucleus in there. That's a smooth muscle cell. There are channels or openings on the surface, sarcolemma, that allow calcium ions to rush in during contraction. So there is a sarcoplasmic reticulum. It does store some calcium, but there aren't any T-tubules and terminal cisternae involved with this business. Let's keep going. Smooth muscle, there are no sarcomeres. There are no contractile units of myofilament arrangement. There is myosin and actin. So I'm just going to write, they do have actin and myosin, but they're arranged a little bit differently. And they're not arranged in that super regular pattern with those sarcomere contractile units. Myosin is shorter in length. It's different. It's a bit shorter in length. I don't want to get into much detail. There are myosin heads, but they occur all along that myosin shaft. So they look differently and they're shorter as well. There is no troponin complex. Remember, troponin is that little protein tab that calcium binds to in order to lift tropomyosin off of the active sites of actin. But there is no troponin complex on the thin filaments. I'm going to say this, the smooth muscle cell it uses something called calmodulin, a protein called calmodulin, to bind calcium. I'll write it out for you. I'll put it in quotation marks. Cal, just like it sounds, modulin, to bind calcium ions. So there is actin, there is myosin. Calcium is involved, but differently, and it's stored. It's not really stored outside of the cell. It comes from outside of the cell during contraction. So the thick and thin filaments, actin and myosin, I'll just write that out, actin, myosin, they're arranged diagonally, not in a long straight pattern like skeletal muscle. That diagonal arrangement allows for smooth muscle, the sheets of smooth muscle, to sort of produce this squeezing action when it contracts. So let me write that, produces. It's like wringing out a towel. All right. Just going to briefly talk about some smooth muscle contractions, and we'll finish this up. So I'll write a header, smooth muscle contraction. So there are things between smooth muscles, connection points called gap junctions. Gap junctions that form from cell to cell in smooth muscle allows the smooth muscle fibers to transmit the electrical impulse from cell to cell. Allows transmission of electrical impulse from cell to cell. So it is the presence of these gap junctions between cells that doesn't require each individual smooth muscle cell to have its own individual nerve ending in order to contract. What that means is if I make up a sheet of smooth muscles cells, fiber cells like this, They occur in sort of these sheet-like patterns, flat sheets. And if I send a neural impulse, let's just say to this cell, oops, there we go. Let's just say to this cell, and let's say this cell. The electrical impulse then can be transmitted from cell to cell, and all of these cells will experience an action potential or an electrical impulse and then contract. So all cells, individual cells, do not need their own specific nerve ending. ATP is still used to energize the myosin head. So I'm just going to write ATP still energizes myosin. That's where the myosin head gains its energy to pull actin. When calcium flows inside of a smooth muscle cell, it triggers the sliding filament theory to happen. So there is still, I'm just going to write, there is still an actin myosin sliding filament action. So it's similar, but not exactly the same. The process is similar, but the steps are a little bit different. I'm just going to hit in one more page and we'll be finished. So here's what happens during contraction. A smooth muscle contraction. Here's sort of a very short Reader's Digest laundry list. First, calcium enters the smooth muscle cell. So let's just say an autonomic neuron has innervated a few of the cells in a smooth muscle sheet. Calcium enters the smooth muscle fiber cell. I'm writing cell, but it's also called a fiber. And I'm just going to put a little bit. So some calcium comes from intracellular sarcoplasmic reticulum. But unlike a skeletal muscle cell where all of the calcium is stored inside, most of it comes from the outside. Some calcium comes from intracellular sarcoplasmic reticulum. Number two, calcium binds to that protein calmodulin. Not troponin, calmodulin. And calmodulin gets activated. Activated calmodulin activates something called myosin kinase. So there's a chain reaction. Activated calmodulin activates an enzyme called myosin kinase. Write enzyme in parentheses so you don't forget. Activated myosin kinase then, so this is really, I had written number 4. Now activated myosin kinase promotes phosphorylation of the myosin head. Remember, ATP is still involved. I'll put in parentheses from ATP. So, but what happens is ATP technically doesn't attach to the head of myosin like it does in skeletal muscle. It donates its phosphate while it's never really attached to the myosin head. So from ATP, I'm just going to write, but the whole molecule ATP never really attaches to the myosin head. It just gives up. Oops, let me spell that correctly. It just gives up its terminal phosphate to that myosin head. Finally, the activated myosin head, number 5, the activated myosin head forms cross bridges with actin in a similar situation, and then it pulls actin. So I'm just going to write activated myosin head attaches actin and pulls. There is no M line, so it sort of pulls in these long wavy motions that produces that squeezing action. Here's the last statement I'm making, I promise. Smooth muscle contractions can also be activated not only by the nervous system, but by the endocrine system, by hormones. So smooth muscle can also be activated. So we can get smooth muscle contractions from both the autonomic nervous system or hormones. That's part of the endocrine system. All right, guys, that wraps up what I wanted to say about chapter 9. I will be back here probably not until after spring break. So we'll probably, most likely, you'll not get a lecture until after spring break and after this next lecture quiz. So if you have any questions, just feel free to shoot me an email. I am going to post an announcement. If you are going to watch this, I'm going to post an announcement. I'm going to push the lecture quiz back one day from Monday, the 22nd of March to Tuesday, the 23rd, to give you an extra day. All right, guys, take care.